Okay. All right, welcome. Welcome everybody to the webinar for today. So we will be speaking on optimizing sample preparation, the best practices with bead-beating technology. And here today, my name is Bobby. I'm the product specialist from MB Biomedicals. And together with me, we have Dr. Sonia Wang. She's the research and development scientist. So together, we will share with you some of the basics of homogenizations and also some of the challenging samples and case studies to tackle your challenging sample types. And during the discussion, feel free to input any questions you have in the chat box. Also let us know if you have any problems with your workflow, particularly on sample preparation. So we will also send you a link in the chat right now. You can access the link to type in your questions during the webinar. So without further ado, let's begin today's webinar. To take you through the content for today, we'll first start with the basics to homogenization, which will include the importance of sample homogenization and a little bit of history and evolution of the entire sequence of homogenization methods. Of course, the common methods of conducting a sample prep. Next, we'll move on to selecting the right homogenization, which includes the overview and the key factors in choosing the right way, as well as choosing the right model. And next, we'll be also mentioning about the right bead types in choosing the right lysing matrix. And last but not least, we'll be introducing the case studies of different challenging samples that may be relevant to your studies. Okay, so again, we encourage all discussions, so feel free to input your questions in the chat box, and then we'll be addressing them at the end of the webinar and discuss with you all together. Okay, let us begin. So first of all, what is homogenization? So the key of homogenization is essentially to be homogeneous. And the word itself means to reduce the substance of what we're trying to achieve to small particles and also to distribute it in a uniform size. So a lot of times when we mention homogenization, it's not only about molecular biology. Sometimes we use it in chemistry, agriculture science, food technology, sociology, and also cell biology. So in the bottom half of this slide, you may see that there are different segregation and different fields of studies that utilizes the technology of homogenization, such as using medicines or herbal pounding, or in dairy industry, which utilizes milk homogenization to separate the fats inside the milk. And also for modern days, a lot of us use it for cosmetic industries, where we apply the cosmetics to our face and body. And last but not least is the lab use of homogenizers, which is here. We have the image of a fast prep 24 machine. All right, so moving on, another concept of homogenization essentially boils down to two important criteria, which is being able to reduce the substance to a very small particle. And not just small, we also want to achieve a distributed in uniform sizes. So the two concepts includes number one being consistency. Being consistency is the ability to achieve same uniform sizes of our samples. So uneven homogenization will leave some of the residuals cells becoming either too big or too small. And that may skew your downstream results, such as your yield or your DNA, RNA purity. And the second concept is the efficiency. So sometimes when we are over-processing or over-shearing our DNA, that may cause certain damages to our target materials, and they could be either our DNAs or our proteins. So again, at the bottom half, we see three photos of different conditions of homogenization utilizing different types of homogenization method. The leftmost being the using the blender. So the first one is using a common food blender where they are disrupting the sample of heart tissue. And as you can see in the image below, the performance result isn't that well because it is utilizing essentially a simpler machine, and it results in a uneven particle, such as the image depicted here. And in the middle, we move on to a better machine or better method. However, it's still not as advanced as a modern day homogenizer. So this is using a motor and pestle, and the sample type that it's homogenizing is muscle tissue. The performance came out better than the blender, but we do see some uneven particles under the microscope. So lastly, on the right-hand side, we see the modern-day homogenizer, such as the fast prep, and utilizing the lysing matrix, which contains the grinding balls inside the tubes. So with the combination of these two, by homogenizing the muscle tissues, we are able to achieve a better and uniform size and also a smaller particle. So um, in summary, this is what we should be able to achieve and what we expect to have for our results. So the purpose of homogenization boils down to three concepts again. Um, I've given an analogy on the right, such as using a coffee grinder as a concept. So for those coffee lovers out there, your coffee comes out perfect every time, where there's no lumps, there's no mess, and there's just only smooth consistency. So it's similar to homogenization, where we really care about the downstream processing to break it down into small and uniform particles. And also for molecular biology researchers, we really want to maximize the yield and quality. And essentially, that has to do a lot with our homogenization process to disrupt the challenging samples, such as cell walls or fibrous tissues. And last but not least, the analysis of tough or complex samples. In this case, we also have soil being a very highly resistant material that is very hard to break down without homogenization. So in the scheme of our homogenization history and evolution, we started in the ancient origins of mortar and pestle, which is a very ancient technique that is very achievable and very easy. All you need is a motor and a grinding pestle that utilizes the force of mechanical. And moving on in the era of 1900s, we started developing glass pestles and tubes. And for a more advanced technology, Potter Alvin-Yam homogenizer was developed, which utilizes the pressurized forced into a glass tubes. So this is more for liquid samples and softer tissues. However, as we advanced through technology in the 1970s, we started developing the bead mill. So bead mill is more of for a industrial field where it's not as advanced as a fast prep homogenizer, but it's used in a lot of the harder samples in industry. Okay, and in the 1970s, we have the sonicator, so which uses the ultrasound to create tiny bubbles and to disrupt some of the proteins and cells. So essentially, this is more for softer tissues and easier to lyse sample types. So for a modern day advanced homogenizer, we have the MP Biomedical's fast prep homogenizer in the 2000s, which we'll be sharing with you on how this benefits our sample preparation. Okay. So with that being said, sample disruption methods has been segregated into two different fields. The first being chemical and the second being mechanical. With chemical homogenization, we also have denaturing and non-denaturing method, where denaturing method utilizes more of a stronger acid or base or even detergent that essentially unfolds the protein. So it's a irreversible method, which is stronger in disrupting the sample. For example, using SDS, urea, or DTT, all these chemicals to break down the cells or our samples. And for non-denaturing, we also have lysozyme, which is a milder chemical that we tend to use it for protein-to-protein interactions where we really want to see the analysis of protein structure. Okay, moving on, we have the mechanical method, which includes complete homogenization or partial homogenization. So for complete homogenization, we uses the bead-beating method, and those are typically for a more tougher or more fibrous resistant materials. And for partial homogenization, we could either utilize nitrogen or by simply cutting it into pieces. Okay, so this is a different common methods of sample disruption, which was just mentioned. We have chemical, mechanical, shocking, and also beating. So again, the chemical method consists of the non-denaturing and denaturing method, which includes different surfactants, detergents, okay, and caryotropes, and also enzymes and other additives. With mechanical method, which we also mentioned, includes the mortar and pestle, the cryogenic way of utilizing the nitrogen way of freezing the sample and also by smashing them into pieces. And of course, the glass homogenizer, which uses pressurized forces and tubes. Next is the sonication, which uses the ultrasound method to break down soft tissues and cell types. And lastly, moving on to bead-beating samples in dentistry, which utilizes the amalgamators and also high throughput homogenizer here. Okay, some of the key benefits, pros and cons for different types of method. With chemical method, and essentially it's the easiest way, and it's also very inexpensive, but it comes with a lot of downside, such as being very time consuming and also prone to introducing molecular sized contaminants. And sometimes in the lab, breathing in too many chemicals will cause potential health hazard. Lastly, it's restricted to samples that are easily dissolved or disrupted. So in summary, there are a lot of limitations for this method. And for a better method, some may use the mortar or pestles. Again, it's very easy and it's very inexpensive. So it's very achievable. So with the mortar and pestle, it's a very low throughput way of homogenization. So it costs a lot of labor and time as well. And because of the working area, we're also introducing a lot of possible potential contamination into our samples. And not to mention that if you're using a glass or porcelain pestle, it's easy to break as well. And lastly, the sample loss due to the adhesion of the surfaces is also a problem. Okay. Next is the French press. This is similar to the glass tubes that was just mentioned. So for this, again, we're always focusing on easy to use and inexpensive, but it always comes down to the the disadvantages. So with the disadvantages, we have the potential introduction of contamination again, being very time and labor and intensive, easily broken. And also for this French press, there's an issue of clogging where your samples are minimized. And it's also limited to usually liquid samples. Okay, for a sonicator, it's a better method since we're using the probe and we're introducing ultrasound techniques. So it's a very fast way of disrupting the cells or proteins, but it's a very poor choice for solid tissues and it generates a lot of heat. So for molecular biology, where DNA and RNA are very prone to heat inactivation, this is very dangerous. And because of the probe usually comes in single-headed, so they may also introduce different types of contamination if it's not cleaned very well. For for the next one, amalgamators, this is mostly for dentistry use, where they use it for tooth fillings. And the main downside for this is the lower throughput. So usually this type of device can only accommodate up to 2 or 3 sample types. So it's very low throughput and also very low efficiency. That's why it's technically not suitable for multi-field researches. Okay, and the next is the bead-beating method. So for this method, it comes in a few different pros, such as being able to accommodate more types of samples, and typically it's more efficient. And the technology, since it's utilizing a different tridimensional homogenization method, so it's more consistent in your samples. And it's also suitable for different types of tissues, yeast, bacteria, soil, and all that, and all those sample types. So if we take into account of different sample types and time and labor time as well, the cost effectiveness is relatively higher than all the other methods. The only thing we would consider for the downside is the heat generation due to the high speed oscillations. But this can be addressed through the cryogenic adapters that we offer, and also utilizing more cooling time. So with that being said, here is a summary of the different homogenization method for you to choose from. But generally, we can see that bead-beating homogenizers are more balanced throughout softer samples up to tougher samples. And it's also the most time-efficient way of homogenizing. So how do we actually choose from the right model if we are looking at bead-beating method? Here are some of the different models that we have, starting with a portable and handheld sample disruption method, which is using the super fast prep 2. So for this, it's a lower throughput, but a portable device that you're able to take into the field and to quickly conduct a sample preparation if DNA or RNA is something that you have to achieve quickly in the field. So the cycle time starts from 600 to 4,800 cycles per minute, and it's also ideal for field-based samples, mainly for soil and feces. Moving on, we have the FastPrep 245G. So this is a benchtop homogenizer. So it is relatively more costly than the super fast prep, but it comes with a higher throughput of up to 48 samples. And it also saves it also saves you money in terms of the amount of sample that it's able to achieve. So the speed setting starts from 4 to 10 meters per second, with usually typical time of completion is around 20 to 40 seconds. So for this model, we currently have more than 5,000 citations and articles published. For a higher throughput need, this model is the FastPrep 96 Pro. So this is able to accommodate up to 192 samples. So essentially, it's a higher throughput machine that only costs you a little bit longer time, since essentially you're homogenizing more samples than the other systems. Okay, it also comes with touch screen and LED light indicators. So for researchers that are limited to budget and they still want to achieve a high quality DNA RNA and does not want to sacrifice too much of their time, they may also choose the next model, which is the Smart Vortenizer. And essentially, this system is a blend of vortex and homogenizer by its name. So it's a compact design for benchtop vortex and homogenization system. The speed range starts from 200 to 3000 RPM, and it is been tested by its performance results that it's effective in extracting DNA, RNA in terms of generating good yield and purity. Okay, so with all the different types of bead beating system that we have, another important aspect of a successful homogenization is to look at what type of beads, grinding beads that you're using. So in this case, we're looking at the different lysing matrices that we have to accommodate to your sample types. From this graph, we're seeing two different axes. The horizontal axis tells you about the aggressiveness, which is essentially the from the left spectrum, we have the lower density beads and also the less hardness beads up until the right spectrum to a higher density and to a tougher and harder beads. Looking at the y-axis, we have angular shape and larger size on top. We also, looking at the bottom, we have the spherical shape, such as using the glass beads and also smaller size beads. So by understanding the different quadrants of this graph, we're able to select the right lysing matrix according to your sample types. So for example, if we are looking into extracting from harder samples such as bones or teeth, you may want to look at the top right quadrant, which is using the lysing matrix A and M, since the characteristics of the beads are more angular in shape, and they're typically larger in size, which is better to pound the hard sample like bones, and to use the angular shape characteristic to really shear down the brittles. Okay, and for softer cells, and tissues or cells, we look at the third quadrant, which is the bottom left. They are typically lower in density, so than less hard than the bones or the teeth. And the shape they come in, the beads that they come in are usually in a spherical shape, such as a glass, and also in smaller sizes. So they're better for softer cells that doesn't overprocess your sample. So we minimize the potential risk of actually either contaminating or degrading your nucleic acids. So again, this is a very intuitive graph that you can refer to in choosing the correct lysing matrix. And to be able to understand the different components of the different lysing matrix, we also provide the composition in them. So with harder beads such as silica or even silicon carbides, you can see that we will tend to select lysing, for example, lysing matrix B or lysing matrix G in this case. So this is essentially to tell you what components are included in our different lysing matrix. So how do we exactly choose the right bead type? There are a few different key considerations to summarize. Number one being, what is your target analytes? Are you looking at DNA? Are you looking at RNA? So to avoid uh, to 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 conduct DNA experiments, usually we avoid harder beads if DNA size matters to you, such as conducting DNA sharing for whole genome sequencing. And if you're looking into softer tissues or even RNA, that are more prone to damage and degradation, maybe select smaller or less abrasive beads in this scenario. Second consideration is looking at the bead size. So smaller size beads are tend to be better for soft tissues and cells, and larger beads are mainly for tougher samples, like what I've just mentioned, the bones, the plants, the yeast, or the teeth sample, the forensic samples. And next would be the different bead shape and material. So in MP Bio, the different lysing matrix comes in either angular shaped or spherical shaped beads that are catered to different resistance samples or normal and mild disruption samples. And lastly, we have different sample volumes for different customers to choose from. So if you have smaller volume, then you can choose the smaller volume versions, including the 2 mLs or the 4.5 mLs. Okay, so we've also summarized a chart for you in this case, and it's segregated into different categories from animal, human tissues to microorganism, environmental samples, plant tissues, insects, and worms. Um, Different types of lysing matrix can be used according to the right sample type that you have. So in summary of what I've just mentioned, when we complete our homogenization, this is the expected result of after homogenization. So we tend to see back to the slide that we just mentioned, we tend to understand that the two important aspect is the size and uniformity. So in this case, we can see that after homogenization, the substance and the particle for all these sample types are grind into small and uniform um, content. Okay, so with that being said, this is a good chance to move on to some of the challenging sample types that may be relevant to you. And we hope throughout this discussion and also the incoming questions, we're able to provide more technical solutions to you. And if you have any questions, feel free, again, to input it into the chat box or the link in the chat so we can discuss with you as well. Okay, so moving forward, the first challenging sample that we like to address is the fungus sample. So with researchers that are researching on fungus samples or trying to homogenize fungus samples, they typically come into a few different difficulties. Number one being the cell wall, since usually yeast cells and fungus family, they have a strong cell wall unlike mammalian cells. And mainly because of the beta-glucans in the cell walls, they form a tough and elastic boundaries inside them, which makes it very difficult to grind it down. And also, the cell behavior itself, like many other cells as well, they tend to clump together when you observe them, and the clumping causes this uneasiness in targeting for homogenization. So here we've given an example of using the typical lysing matrix Y, which is very suitable for either yeast, mold, or algae. So essentially the fungus family. And its component includes the 0.5 millimeters zirconium beads, which is a all-around and good bead for fungus family. And lastly, the shape itself is in ball shape, so it's not an angular shaped bead, which tend to be better for plant cells and fungus cells. So with that being said, this is a good chance to introduce Dr. Sonia to give us some insights on the first challenging samples that we have and some of the case studies that cater to this type of challenging sample.
Hey, thanks, Bobby, for the very informative session where he actually introduced about fast prep beads-beating homogenization as well as some of the basic rule of thumb that guides you in selecting the lysine matrices. So, like what Bobby mentioned, the first one that we want to address is especially when you have a very little quantity of the fungus, it makes it even more difficult, just like this case. So in this group of researchers, they are trying to extract fungal isolate from um, different strains, different fungal isolates. Some might be, the culture amount might be lower, some might be more. But what they are trying to do is they are trying to get homogenize it in a way that they can get the proteins to do the protein profiling. So some of the key challenges would be uh, the robust and chitinous cell wall of the filamentous fungi, which makes it which makes it an additional barrier to efficient homogenization before we can get our hands on the proteins, and especially in stringent or extreme experimental parameters, they have no choice but they can only work with low biomass fungal sample. So this low biomass build-up actually makes the whole homogenization even more tricky if they want to get a good, handsome amount of protein out of this homogenization. So this group of researchers actually use our FastPrep 24/5G to actually do a 6.5 meter per second homogenization for 60 seconds twice with 5-minute interval for the first lysis buffer, followed by single homogenization for the second type of lysis buffer, where they were also testing the different lysis buffer recipes. And the beads they used were zero point 0.1 millimolar zirconia beads. And a similar one that we have and we could offer is lysine matrix Y. And they tested on different fungal isolates, and this is just a brief summary of their result. Meaning they have selected this tris magnesium chloride 3 buffer system, which is their best lysis buffer among their tested lysis buffers, and they paired with fast prep homogenization with zirconium beads actually worked well for all of their fungal cultures. And the outcome was they got a protein concentration above 1 milligram per mil from lyophilized amount of only as little as 5 milligram. So it goes to show that fast prep with its three-dimensional uh, movement, it actually built up the vertical force as well as the angular force within the tube itself that creates good compaction, as well as with the minor particles like zirconia beads in its small size. It will be brought together within the fluid vortex during the homogenization, and in this way it creates the shearing effect to fully homogenize half samples and for us to get the most out of it, even though it's as little as this fungus 5 milligram in this case. So in this paper and in this group with researchers, the with this complete homogenization, they have successfully proceeded with the downstream applications of protein extraction, proteomics analysis of this fungus species stated here, as well as protein profiling of samples or organisms which are difficult to cultivate or sparsely obtain.
So, Dr. Sonia, I'm seeing a lot of different fungal species that's been used. So essentially, I think we're able to say that using the zirconium beads provided in this lysine matrix, it's an all-around solution for all these types of fungal species, right? To,
Yes.
to mainly to address the two challenges that you've just mentioned, either the low biomass or the robustness or the chitinous of the cell wall.
Yes,
Yes.
precisely.
Okay. All right, thank you, Dr. Sonia. And moving on to the second challenging sample that we have, which is a lot of the users are conducting, which is the forensic samples. And as we know that forensic sample is a wide and a big, broad range, includes a broad range of sample types, right? So, for example, we have hair, we have bones, um, we have cartilage, and all these are included in the range of forensic samples. So with that being said, they all poses different challenging features. So for example, bones, they are extremely hard. So you would have to really break them into pieces and to be able to extract from the minimum DNA that they have. And also for hair, they usually consist of keratins, and the DNA contents are usually in the cell roots. So it's very low biomass as well. And lastly, the shape of itself is also a challenge. So bones would require a pre-preparation. So by either using the nitrogen method to freeze it and to break it down into smaller pieces, then you would proceed to conduct homogenization, which adds another layer of difficulty there. So with that being said, I think we can also address this type of challenge by utilizing the lysing matrix S, which is very suitable for forensic samples mentioned just now. It includes the bead type of 3.1 diameter of stainless steel beads. So this is a strengthened bead and tube, which is in ball shape. That is very useful and uh, very suitable bead type for forensic samples. And again, with that being said, I think Dr. Sonia can introduce us again some of the challenges for this particular type of sample as well.
Yes, sure, Bobby. Yeah, so the example of case study that I've chosen for this challenging one would be a paper that studies on, bovine cartilage, where they are trying to get good quality of RNA from bovine cartilage because they are studying about the lubricant as well as if they introduce externally a lubricant for an inflamed joint. So would it actually help in upregulating some markers that actually reduce the friction for example, osteoarthritic patients? So some of the key challenges, just like Bobby has mentioned, bone samples are tough. Bone or cartilage, they are actually tough. So they create even a stronger barrier against sufficient homogenization. Moreover, because bone is made up of the different type of minerals, so this bone powder further trap the nucleic acid, whether it's DNA or RNA that researchers are interested in. So this creates the problem or challenges that we face when we want to get RNA from these samples. And also, uh, it is hard, especially for RNA extraction, when we want to take care of the integrity as well. That means we don't want to pound the sample too much to the point that RNA is degraded. So another challenge here would be to achieve a complete homogenization, yet maintaining downstream compatible RNA, meaning your RNA is still usable for your downstream applications. So in this case, again, FastTrack24 5G has helped this group of researchers to do a just nice homogenization, which is 15 seconds for 10 cycles with 2-minute interval on ice using Kaizo buffer, where it has actually helped them to get a good RNA integrity, a good RNA yield and integrity sufficient for their outcome, which is they did a qPCR on the recombinant proteoglycan 4 protein, which is their marker of interest in this case. So like what I've mentioned, this RNA has proven to be usable even after the complete homogenization because the researchers have successfully done cDNA synthesis and qPCR. And in this case, it has helped them to identify the pro and anti-inflammation markers and mRNA expression. And this would actually contribute to a bigger goal of personalized medicine as well as new drug discovery.
Okay, so to add on to Dr. Sonia's case study, I think this gives us a good chance to talk about the further challenges that our customers are facing as well. So with forensic samples, um, we usually tend to have difficulties like over shearing and also over processing, which generates a lot of the heat. So for heat, how do we actually address the concern for that, Dr. Sonia?
Yeah, so Bobby has raised a very good question, which is RNA is very heat sensitive. And how do we address that? Of course, just like what you have probably seen from this case itself, they actually conduct an ice incubation interval for a few minutes between the homogenization cycle. Of course, another way is we also have a cool prep adapter for all of our adapters, where you can actually cool the sample with surrounding dry ice during the homogenization itself.
Okay, thank you. I think with that being said, we can also use this chance to address one of our customers' question. I think he is from Korea. So he's saying that I'm conducting experiments with samples that are not a regular hair, but it's more of a bristle-like sample, such as pig bristles. And because of this, he uses larger bead size, and he's worrying that if we're prolonging the beating time, it may damage the sample. So can we give some suggestion or efficient method to process this coarse hair sample, um, while also maintaining and minimizing the damage? Yeah.
Yes, Bobby. Pig bristle is a very interesting sample. Yes, of course, there's a concern. I'm not sure whether this person is interested in RNA or DNA, but regardless, of course, we want to maximize the DNA or RNA or other macromolecules that you are looking for from the sample. We want to maintain its integrity as much as possible. So in this case, I would actually recommend using lysine matrix A or M, because in this case we handle both sides well. With A, actually they lysine matrix A has a garnet material, garnet shards, where it has a angular shape, or rather a rougher surface, and it's in smaller size, where it in irregular shape as well, where it can actually increase the shearing force. And also with the zirconium beads of M, it can actually create the cascade compaction that I mentioned through our fast prep vertical and angular movement. So in this case, by introducing both compaction and shearing forces, definitely you could actually achieve the homogenization within a shorter period of time, and that will actually save you from degrading whatever macromolecule of interest that you want to take from the sample.
Okay, thank you, Dr. Sonia. I think hopefully that explains the question that our customer has. And again, feel free to input your questions in the chat box so we can address directly. All right. So yeah, I think moving on, we'd like to address the last sample that we have, which is also a very highly concerned sample type. So this one is the tough and fibrous tissues, so such as muscle tissues, heart tissues, or even plant or fatty tissues. So these type of tissues are usually having a much more structurized cell membrane. So with plants, they're usually fibrous and also contains a lot of carbohydrates, in especially lignins in stems and roots. So this makes them very hard to disrupt. And as for fatty tissues, and it's very greasy, so the consistency can also affect our outcome as well. So I believe a lot of our users are extracting from these types of tissues. So in order to address this, we recommend the lysine matrix A, which Dr. Sonia has just mentioned in her previous case study. So this type of lysine matrix bead is very suitable for either plants, tissues, or even environmental samples like soil and feces. So because of the beads type it's containing, it has the zirconium sphere and garnet flakes, which is more of a all-around angular shaped beads that's able to tackle all these tissues, all these samples above. Okay, so again, with this type of tissue sample, I think we can also mention one of the case studies that's been brought up by Dr. Sonia again.
Yes, this is definitely an interesting tissue, where interesting sample, where it's also considered challenging, because so the one that I'm talking about now would be tough fibrous tissue which is muscle, that this group of researchers actually wanting to extract RNA from this tough and fibrous tissue. So what are the key challenges for tough and fibrous tissue would be, of course, they are fibrous and hard. So it hinders complete homogenization. And also, again, it's a similar challenge that is shared by previous case study. We want to achieve a complete homogenization, yet maintaining good RNA integrity. In simpler words, meaning we don't want to pound it too much, we don't want to grind it too much to the point that we degrade the RNA. So this group of researchers actually use our fast prep 24/5G to actually do 6 meter per second for 40 seconds and 4 cycles and 5-minute interval on ice. Again, you can see that the ice incubation interval is very important, especially when you're dealing with RNA. Using these buffers, lysis buffers, and the beads or lysing matrix used was actually 0.5 millimolar silica or zirconium beads. So in this case, the fast prep 24 homogenization with these silicas and zirconium beads was able to achieve more consistent homogenization if I bring your attention to the left side of the graph, where you can see that across different sample types, whether it's visceral adipose tissue or subcutaneous adipose tissue, SCAT, or liver and muscle, we can see that fast prep 1 actually gave more consistent RNA yield. And then on the right side, you can see that for even tough and fibrous tissue like muscle, after using fast prep 24 to homogenize or break it into smaller pieces, we still could achieve complete homogenization and giving subsequent RNA of high yield and high integrity, giving a high RNA integrity value as high as 9.1, as you can see here. So again, this proven that FastPrep 24/5G, we could achieve good homogenization at the same time maintaining good RNA integrity. And this group of researchers have actually continued on with their work, and the RNA was actually proven usable for their RNA sequencing and microarray. And they've also done transcriptomic profiling of these metabolic tissues. And again, this has contributed to a bigger goal of personalized medicine as well as new drug discovery research area.
Okay, thank you, Dr. Sonia. So I think with that being said, this is also a good chance to introduce some of the questions that we have, especially for tough and fibrous tissues as well. So I think some of the questions are coming in and we are receiving that. So thank you very much for the question. But before that, I think we can do a little bit of summarization on the different sample types that we just mentioned. So including fungus cells, forensic samples, and tough fibrous tissues. Especially for the first one, fungus cells, the potential challenges that we're facing includes the robust and chitinous cell wall, and also the low biomass. Of course, also if you're using sonication or pestle mortar, typically that is not the best option here. So the potential solution that we provide is by using the zirconium beads and the silicon carbide beads, which is essentially a harder material that is able to tackle the hard cell wall. And also, it's important that we look into the solubilization in our buffer system, such as using the tris buffers or the TTs or urea to pair with the lysine matrix to lower the heat generated factor. And lastly, of course, to minimize any protein degradation by using intermittent cooling, using the cool prep adapters, or even simply by taking them out and cooling for a few cycles. So yeah, next with forensic samples, the potential challenges that we're facing also is the keratins in hair sample and also mineralized matrices in bones. So these are the top challenges that we face. And to address that, we can either by crushing it into smaller pieces, but a better way is to select the right lysine matrix for tougher beads to tackle these harder and denser samples, especially when you're dealing with bone samples where it's bigger in pieces. you'll tend to need a bigger beads to pound it in pieces first. Then by selecting a more angular shaped beads to minimize into smaller fragments. Okay. And the last is tough and fibrous tissues, where we're facing different visceral fats, such as lipids, and also fibrous nature, for example, the collagens. So a lot of times when we're extracting from one type of tissues, it comes with a variety of complex physical properties. So these are all the challenges that we're facing, particularly for this type of tissue. And again, the potential solution that we can provide is by using a higher concentration of a buffer system, such as the guanidine-based salt, or even beta-ME for cell lysis. And in terms of homogenization, we can select lysing matrix A, so which is a versatile and all-around beads that's able to accommodate multiple sample physical properties. So in summary, we've addressed the different challenges in different sample types. And back to the best practices with our bead beating technology. First, we have to take into different considerations. First being to understand your sample throughput and to select the right homogenizer. So for instance, if you have a lower throughput, such as one or two samples, maybe super fast prep is ideal for you. And as we go higher in our capacity or sample volume, we would like to choose a more capable homogenizer, such as the FastPrep 24 for maximum of 48 samples. And for even higher throughput, you may also go for the FastPrep 96 Pro. And again, for budget limited users, the Smart Vertinizer, essentially a blend of homogenizer and vortex, is also suitable for you with a sacrifice of a little bit of longer time. But again, it's able to achieve the optimal DNA yield and purity as well. Okay, so moving on to the right side, the second consideration is to look into the different properties of lysing matrix and beads. So with that, again, we're looking at the target analytes. Is it DNA? Is it RNA? Or are you looking at proteins? With that, we're looking at the different bead size as well. So we just mentioned, usually for smaller beads, it tends to work better with smaller cells, softer tissues. And for larger beads, it tends to work better with the tougher forensic samples. Okay, and for the next part, which is the bead shape and composition, again, we take into account of the different composition, either angular shaped or spherical shape of the beads. And lastly is the sample volume. So how much throughput are you using? Okay, so before we end the webinar, let's address some of the questions that our users have. Okay, just a moment. Okay. All right, let's look at some of the incoming questions. The second question that we have is from Annabelle. Thank you for the question. So I think she's saying that I would like to receive training content, either as YouTube upload or recorded versions. Okay, so we will be posting this in the near future. So we'll provide a YouTube link on this. And she's also asking that she's been using SpinEasy RNA kit for bacteria, but she keeps on getting a very degraded and minimal RNA. So how can we troubleshoot that? So her sample type is gram-negative cell, like E. coli, incubated in minimal media and yeah, chitins. Okay.
I think for Annabelle's question, right, we probably would like to know more whether the starting material was enough. And of course, with that being addressed, I mean, if you were to provide us with that information, we will be happy to know, because starting amount will actually affect your extracted RNA yield and quality, definitely, because if the starting amount is not enough, then probably you will get a low RNA yield, which is also directly related with RNA integrity, because most of the time, if you don't extract RNA with a high enough yield, then probably it will affect its integrity, and probably you'll get more degraded RNA as well. But in this case, what I would speculate is that probably you might not want to over lyse or over homogenize, because gram-negative bacteria is comparatively easier to homogenize than gram-positive bacteria, because of the absence of the peptidoglycan cell wall. So in this case, probably you have to tweak the homogenization speed and time to make sure that you get just enough RNA yield while maintaining it in its good integrity state. Yeah.
Okay, I think we can also address the minimal RNA that she has, right? So by possibly by also lengthening the binding time, that could also help in achieving more yield, and also possibly by lowering the elution volume that she has. Okay. So again, thank you, Dr. Sonia. Let's move on to the next question we have. Okay, let's see. We have Gazel. Okay, so thank you for the question. So he's saying that I have groundwater sample, which is very oily and dense. What kind of lysing matrix should I use for them? So I think typically for oily samples, especially in water, we use lysing matrix E. But of course, one of our kits, which is Spin Easy kit for water, it utilizes the filter membrane as well. So it's very important that we separate the oil and the water to essentially get a good yield and, of course, a good purity in this case. So anything we can add on to, Dr. Sonia?
Yeah, no further comment, because I agree with using lysing matrix E as well, because it can handle a wide range of environmental samples like soil or feces or wastewater, or yeah. So when it includes, I mean, wastewater actually somewhat meets your criteria, because it could be very oily or dense, or with different kind of foreign particles. So I will also suggest lysing matrix E.
Okay. All right. Thank you. Thanks for the question, Gazel. That is a very good question. Let's move on to the next one. Zara. So Zara has a question. She's saying that I'm working with fungi and was wondering if the aggressiveness of our fast prep could impact the enzyme extraction efficiency or might possibly degrade the enzymes.
Yeah, that's a very interesting question, because you are looking at only enzymes instead of DNA or RNA extraction, meaning you can probably do with you can probably go with not so harsh homogenization. So in this case, probably large size beads would be sufficient to create a compaction where it breaks open the sample that you have, which is fungi, that is just sufficient for you to get your enzyme. So this is similar to my case study one when we talk about fungus, just like proven by that case study as well. They have gotten successfully gotten the proteins. They are not just looking at one enzyme, but they were looking at proteins, and they have profiled the proteins after using fast prep to homogenize and get the total proteins from these fungal isolates of different strains. So definitely in this case, if you are concerned of the extraction efficiency or enzyme degradation, you probably would want to go for a medium speed, and then maybe 1 minute, and then put it to rest in between with intermittent rest, and then you do another cycle. And if you do not get enough yield or enough enzyme yet, then probably only you increase the cycle progressively. That will probably help with your optimization work in dealing with this fungi.
Yeah, so Dr. Sonia, I think this is essentially a lot of times this is more of a trial and error. So I'm assuming that he or she hasn't done this sample disruption before. So I mean, we could start with starting from a lower homogenization speed, right? To test it further.
Yes.
Okay. All right. So we have one more question, and please keep the questions coming so we can address it directly. So with that being said, we have one more. So the question is, we've discussed about the animal and human tissues. What about plant tissues? So how do we deal with the plant tissues? How do we treat it?
Garnet shards as well as the ceramic beads. Uh, and and yeah, so lysine matrix A actually gives a good balance between the compaction that we need as well as the shearing force that we need to tear open the the plant tissue for us to get the most out of it, whether it's protein or RNA or DNA.
Yeah, I think also sometimes when you receive plant samples, they come in larger size, right? So sometimes nitrogen freezing it and by breaking them down in smaller pieces before you actually homogenize them is also very important in this case. So again, just like we mentioned in the case studies, plant samples have very fibrous nature and also they are tough in their cell wall. So whatever Dr. Sonia just mentioned is something that we can take into consideration. So I hope that addresses your question. And let's see if we have more questions coming in. So again, you are looking at two QR codes here. So if you need any help, please visit the QR code to the left where please visit the QR code to the right where we have the technical support by scanning the QR code on top. And also to the right, we have Fast Prep demo request form. So by scanning that, you're able to request a free sample demo of our Fast Prep machines, either being 24.5G, the higher throughput ones, 96 Pro, they can all be demoed. Okay, so let's wait a bit for any more questions in the chat. Okay, let's just give it maybe a few minutes, maybe 1 minute, to see if any questions are coming in. So I think this is a good chance to also address that we provide this technical support for you. If you're facing any difficulties in your extraction, it doesn't have to be homogenization. It could be during your molecular biology when you're facing low DNA yields or bad purity. I think we have this team to fully support you. So all you have to do is to scan the technical support QR code. So it does not have to be only catered to homogenization. Okay, let's give it maybe another 30 seconds. Any questions in? Okay, I think we have one more question here. So the question is for the pig bristle samples mentioned earlier, they're used for cortisol measurement, and the customer is concerned about the damage due to long milling time when we're using a larger bead. Would switching to lysine matrix A or M alone be enough to resolve this issue? Um, I think this has to do with also the time, so not just the beads itself. So if we are prolonging the time, then whatever beads you're using doesn't really affect. So I think this boils down to the processing time as well. Dr. Sonia, any comments on this one?
Yes, I agree. I fully agree with Bobby because, of course, changing it alone without optimizing the speed and the time, it wouldn't bring you I mean, it wouldn't be a good optimization, because why I would recommend A or M is actually, in this case, the concern was actually long milling time. And why A and M is because it could actually put, like what I said, it could give you a good balance between the compaction that we need as well as the shearing that we need. So that is contributed by the different sizes of the beads or material. So in this case, with a better choice, or with somewhat like a matrix that can have a good balance of both worlds, then the next step is for you to optimize the speed and the time of homogenization in order to achieve your specific objective, which is the cortisol level that you would want to get. Yeah.
Okay, thank you, Dr. Sonia, and thank you for the question. All right, so I think that is everything. If there's no more question, if there's more question, feel free to input it into the QR code. We'll be happy to help after this session. And if not, I think we can conclude the session. But before that, please help us improve our webinar by scanning the QR code again on the feedback survey. So this will help in enhancing our future content as well. So I'll give it a minute for everybody to scan. And I think we can end the webinar for today. So thank you very much for attending. And I hope everybody has learned something from this session. Okay. See you all. Thank you.
Thank you.